Thank you.
Okay. Good morning, everyone. So before I begin, I would like to mention that I am not a clinician, but I'm a researcher working closely with clinicians on this project. And as you can see, the title of the project, it is very complicated, I know. That is CAM Co-Culture, a translational model for drug discovery in metastatic uveal melanoma. So I hope to explain its key aspects, and I will make it easier for you to understand. So as a researcher, this is our overall research pipeline, which starts from the idea, developing an idea, to treating a patient. So it has three different stages. Under discovery part, we work in labs and we work on animal models. We do drug screening. And then in development part, the drug goes through clinical trials, phase 1, phase 2, or phase 3 clinical trials. And then it goes through regulatory approval, and then it could be chosen for like a therapeutic option. So all these three stages, it takes around 10 to 20 years, be it any cancer or any disease. So I would like to start this with a question that what is actually a cancer? So our body, it is made up of like more than 30 trillion cells, and it makes whole tissues and organs inside our body. So normally cells in our body, they grow in a very controlled mechanism, and it makes our body in a very healthy state. But in cancer, things can go wrong, and our cells, they can grow in a very uncontrolled fashion, and then this can cause the formation of cancer cells, what is known as tumor. So here it is in sorry. Yes, so this is an example of an eye cancer, as we all are familiar with, that is choroidal melanoma. You can see here the clump of cells making a tumor inside the eye. So what provides the instructions to the cells that how should they behave, when they should stop dividing or when they should start dividing? The answer lies in our genes. So genes provide instructions that controls the behavior of our cells. But in cancer, the mistakes appear in these genes, what is known as genetic mutations. So because of these genetic mutations, the tumor can develop in our body. Now, the basic aspect of the cancer is the tumor microenvironment. It is basically the environment around the tumor, which consists various types of cells that are stromal cells, immune cells, or blood vessels. So because of this tumor microenvironment, the tumor can spread from one part of the body to the other parts of the body. So, and yes, the tumor microenvironment can also help the tumor to hide from our immune system. So this can influence the response to treatment of the patient, but it also expands the treatment options that we can do research on. Now, as we all are individuals with our own characteristics, similarly, all patients are different because they have different genetic alterations and their tumor microenvironment is different. So studying those genetic alterations and specific tumor microenvironment can help doctors to choose the treatment that most likely work for them. So we all know there are various therapies in uveal melanoma. The most common is chemotherapy. So in chemotherapy, when drug is administered, it attacks the cancer cell and it can cause cell death, cancer cell death, and tumor shrinkage. But with cancer cells, it can also affect the normal cells which are present around the tumor. So it can attack the healthy cells. That's why we can see some of the side effects such as nausea, hair loss, weak immune system, and other side effects like vomiting. So that is why there is low response rate in uveal melanoma with chemotherapy, and that is why it is not considered effective for treatment. So another approach instead of chemotherapy is chemosat. So in uveal melanoma, this is given as liver-directed therapy using melphalan drug. So this chemosat has shown better response rate and lesser side effects in comparison to chemotherapy. Now, next therapy is targeted therapy. So unlike chemotherapy, it is very specific. It attacks a particular molecule inside the cell, which is responsible for the cancer cell growth. So here you can see the red dots. They are, for example, the molecules inside the cells, which are serving as targets for the drugs which are we using for targeted therapy. So after attacking these molecules, they can stop the growth of cancer cells and can cause cell death and tumor shrinkage. Because targeted therapies are very specific, they do not attack the normal cells around the cancer cells. That's why they have better response rate, and this is an example of the combination drug Darvo and crizotinib, which is under phase 1 clinical trial in patients. Now next, which is very important, is immunotherapy. So it basically activates our immune system. So when this drug is administered, it switches on our immune cells, and this immune cell can target the cancer cell and causes its death. So TEBI, which is the most common form of immunotherapy, has been used to treat advanced and metastatic uveal melanoma. When I talk about metastatic uveal melanoma, it means the primary tumor from eye, it can travel through blood vessels in the various parts of our body. So it majorly takes place in liver, that is 90%, and can be also rarely seen in skin, bone, or lungs. So now how TEBI works. So this is the cancer cell, and this pointer is not working. Okay, so this is the cancer cell, and our immune cell is in inactivated state. So when the TEBI is given, what it does, it brings the cancer cell and it activates the immune cell, and it brings the immune cell in the close proximity of cancer cell, and ultimately it kills the cancer cell. So this drug, it has shown the increased overall survival in patients. That is why NHS has announced the availability of this drug for 100 of patients. The only limitation is it is only effective in patients with HLA-2 positive cells. Then what about those patients who are HLA negative, or those patients who have fewer treatment options? So now my work lies here under the discovery part, where I am working on preclinical models. So preclinical models are experimental system which we can use to study disease or any cancer. And we can also do drug screening in those preclinical models before testing treatments in human. So why do we need preclinical model? Firstly, it is very similar to the cancer which is actually present in the patient's body, because it is three-dimensional in its structure, it has blood vessels, and it has tumor microenvironment. So the more complex the preclinical model would be, the more confidence the result will translate into human patients. For example, if I'm working like on normal established cell lines, that is less complex. That won't mimic the tumor which is present inside the patient's body. But if I'm using on patient samples, then it would replicate or mimic whatever is present inside the patient's body, and we can do drug screening on them, and that will be more effective. So what we can study with preclinical models, that what makes a cancer cell more likely to spread, and what makes cancer cell more likely to respond to current treatment. And we can also identify clues to help design new treatments, and then it can be used as drug screening. So the gold standard for studying uveal melanoma has been to use mice. So what we are doing in Liverpool is developing an alternative preclinical models that utilizes eggs. So why are we using eggs? First, it is very cheaper. And second, when we work with mouse or rats, it takes around months to get the results from them. But with eggs, it just takes 14 days. And third, also, we are trying to make our study more efficient and more ethically correct. So here are some images from our lab. So here you can see the eggs which we can buy from the supermarket. And behind the eggs, you can see incubators. So in these incubators, we keep the eggs while we do the experiments. It maintains their standard conditions like temperature, humidity. And then here you can see when we do the experiment, we create a hole in the egg. And by creating the hole, we can implant cells or patient samples and can also do drug treatment inside the eggs. So coming back to uveal melanoma, as we know that it mainly metastasizes with in liver, but we don't know that why and how they interact with liver cells. So what I am doing is I am mixing cells, that is uveal melanoma cells, which are already established cell line, with liver cancer cells. These are also already established cell lines. I am mixing them together, and then I'm implanting them in the egg. After implantation, after 7 days, I got this tumor. So in this tumor, you can see it is a completely 3D structure. There are tiny blood vessels which mimics the tumor microenvironment of an exact tumor. Now, how would we know that in this tumor, what are cells and what are liver cells? So what we have done, we have tagged the cells with green dye, and we have tagged the liver cancer cells with red dye. So now you can see in this egg, you can see the green fluorescence, which represents the cells, and the red fluorescence, which represents the liver cancer cells. Now we dissected out the nodule from the egg, and this is what we got, the 3D structure of the nodule having both the cells together. So, as I have mentioned, we have used liver cancer cell lines with cells, and we successfully developed the tumor. But what we actually want to use normal liver cells because it will be obtained from the patients, and it will closely mimic the patient tumor microenvironment. So next what we did, we mixed cells with the liver cells obtained from the patients, the normal liver cells, because metastasizes to the normal liver, not the cancerous liver. So this is the tumor we got with cells and the normal liver cells. And this is the tumor inside the egg. This green color, it represents the cells. And then we dissected out the nodule from the egg. And you can see that with normal liver cells as well, we got a good nodule with blood vessels. Now, to identify that how are they interacting with each other, the liver cells and cells, we sliced this tumor into very thin sections, like the slice of a bread, and then we stained with dyes. And then you can see these light pinkish color are basically normal liver cells obtained from the patients. And these are the cells which are around the liver cells. So you can see that both normal liver cells and cells are interacting with each other. Now, what my next step is, after developing realistic tumor, our aim is to take the liver cells from both HLA positive patients and HLA negative patients, and then we will test it with absence or presence of TEBI, and then we would understand that why HLA positive patients, the TEBI is effective why in HLA positive patients but not in HLA negative patients. After understanding these differences, what we can do that we can design new drugs which could be effective for in HLA negative patients or for those patients with fewer treatment options. So now, we have developed the model with cancerous cell lines. We have developed the model with normal liver cells, and cell lines. Next, what we want to do is we want to develop a model with precision cut liver slices. So these are very thin slices from the liver metastatic patients that would already contain the cells that have spread there. So this approach will let us study tumor exactly as it exists inside the patient's body. So by using these liver slices from patients, our goal is to use HensEggs as patient avatar. So for every patient, we will be able to study their unique tumor, identify their specific genes. We can predict the best targeted drugs and how they are responding. So this will make this approach will bring us more closer to personalized medicine, and it will give every patient the best possible chance. So that's it. I would like to thank all my mentors and all my patients who have contributed in my research. Thanks a lot.